Hello again, everybody. So this is our second video that I want you to watch in place of lecture today, and this one's really going to contain the bulk of what I intended to lecture on today. All right, so the big topic of this video is DNA repair, and so just as an intro into this one, we'll kind of cover some kind of basic facts about error rates and mutations in genomes. So studies that have looked at sort of the overall replication error rate of DNA polymerase during replication indicated that there is an error. DNA polymerase makes an error about one base every 100,000 to a million bases during replication. So as I mentioned on Monday, DNA polymerase isn't perfect, it makes mistakes. And so this comes out to about 3,000 errors per genome per replication cycle, right? So this may seem like a pretty low error rate, but remember our genomes are very large, 3 billion bases. So if you apply this across the entire 3 billion bases, this ends up to about 3,000 errors per genome, right? So that's quite a few errors, okay? But if you actually look at the actual mutation rate in organisms, it's much lower, okay? So the actual mutation rate's about 1 mutation per 100 million bases, right? And so with some kind of calculations, this comes out to about 60 errors per genome during gamete formation, right? So this, if we just kind of look at this as at the face value here, I know these are kind of different units here, but let's just kind of look at the numbers here. This number here, 60, is much lower here than this number here. And this is actually largely true. The actual mutation rate in organisms is much lower than the error rate, right? So that's kind of our driving question here. Why is the mutation rate lower than the error rate in organisms? And this kind of it covers all organisms, going from bacteria all the way through humans, right? And so there are two main mechanisms that we're going to talk about today. The true answer to this is there are a lot of reasons why this mutation rate is lower. We can't talk about all of them, but I want to kind of cover two main reasons why this mutation rate is lower. The two reasons we're going to talk about are first, DNA proofreading, and the second one is DNA repair, right? So we're going to end up focusing on DNA proofreading here first, right? Because I think what this does is it illustrates a really interesting kind of property of DNA polymerase, and that it's actually a very complex enzymatic it's a complex enzymatic complex, right? It actually contains multiple functions. We focus largely on DNA polymerase's role in DNA synthesis, but it actually contains multiple functions that allow it to do many different things, right? So let's focus on DNA proofreading. And I think it's okay to kind of think about this in rough terms as basically it's spell check for DNA polymerase. It kind of has the ability to, as it's synthesizing DNA, detect that something is spelled wrong or there's some kind of error, pause, eliminate that error, and then kind of continue and put in the correct base. So this is, you can think of this as spell check for DNA polymerase, right? So this is really, it's an inherent property of DNA polymerase. Okay, and I would say the big key to this mechanism is that this occurs during replication. And this will contrast with the next mechanism that we talk about. Okay? And so really, because this is a property of DNA polymerase, this really depends on the ability of DNA polymerase. Okay? So DNA Pol, it can detect incorrect base pairs. Okay? And so if, as DNA synthesis is occurring, the wrong base pair gets incorporated, DNA polymerase can detect that, it can pause, it can eliminate that wrong base, and then actually resynthesize to fix that mistake. Right, so again, this is an inherent property of DNA polymerase, and it's because DNA Pol can detect incorrect base pairs. So let's actually get into that a little bit and draw out the mechanism here. Okay? So let's say we've got a stretch of DNA. Let's say we've got 3' to 5' stretch on the bottom here. Okay? And let's say that this is a template strand down here during replication. Okay, so this is during replication. Okay. So DNA polymerase is synthesizing DNA this direction on this new strand in the 5' to 3' direction. Okay, and let's say that there's an adenine base right here. Okay, and for our example, let's say that in this case, what happens is DNA polymerase, we'll put that one in purple, and it's kind of latched on here. Okay, and let's say in this case, a guanine base comes in here in place of the correct base pair, which would be a thymine. Okay, so as you know, that this is an incorrect base pair, this GA base pair. Okay, so here is our issue. An error has been made during synthesis, the wrong base has been incorporated. Okay, now this will kind of go back to some things we've talked about on Monday. If you have the incorrect base pair present there, that actually creates the wrong shape in this very specific kind of pocket where DNA poly is interacting with the DNA, okay? And that's effectively what DNA polymerase can detect. So this GA, this incorrect base pair, actually conforms the wrong shape, okay? And that's what's being detected by DNA polymerase. As it goes along, it can kind of detect the shape of the base pair that's being made. If it's correct, and it kind of fits into the pocket of DNA polymerase, detects that and continues on with synthesis. In this case, we've got the wrong base pairing matchup here. That creates a slightly different shape here. DNA polymerase can detect that. Okay, so this signals to DNA polymerase to pause. So DNA polymerase pauses, and then that engages a secondary enzymatic property of DNA polymerase. What that engages then is it engages the exonuclease activity of DNA polymerase. So DNA polymerase also has what's known as 3' or 5' exonuclease activity. Okay? And so just let's walk through this a little bit. 3' to 5' implies a directionality for this function. And then the exonuclease is the actual function, right? So there's our ASE suffix. And so it's going to be an enzymatic function of exonuclease, or sort of degrading nucleic acids from an end. That's what exo means in this case, right? So DNA polymerase now, at this point, it's paused. It can actually engage exonuclease activity, and it'll actually go backwards, right? Okay, so what it's going to do is it's going to go back in this direction on the strand, and it's going to eliminate this G. Okay? So we'll go back to we'll redraw our strand. Here's our 5' end of the new strand. Here's our template strand on the bottom. Okay? We know the next base that it's trying to match up with here is the A. So in this case, DNA polymerase actually chews back where that G was and kicks the G out. Okay? That's the proofreading. Okay? And this is because of this 3' to 5' exonuclease activity. Kind of chews back this direction a little bit, kicks out the G, and now it's poised and ready for the next correct base pair to come in. Okay? So we'll just kind of make this a little bit smaller down here. And now we have our 5' end of the new strand. Here's the 3' end. Here's our template strand on the bottom. We have an A. Okay, and now that that wrong base is gone, the correct base can come in. Okay, in this case it's a T. It can be inserted right in there, and now DNA polymerase can actually continue with its normal 5' to 3' polymerase activity. Right. So this is DNA proofreading. DNA polymerase has the ability to do this to detect when the wrong base comes in. It happens, but DNA polymerase can detect this. I would say an exception to this, we talked about this on Monday, is if there's a tautomeric base pair interaction formed. Remember, those are the ones that can actually do mimic the shape of a real base. And so that's how these errors can actually sneak through the cracks of DNA polymerase. This is really one that can recognize actual incorrect base pairing here, right? And then just illustrating that DNA polymerase is a very complex enzyme. Okay? It contains multiple functions. Not only does it contain this sort of 5' to 3' synthesis activity, but also has some components that can engage in 3' to 5' exonuclease activity, right? So that's DNA proofreading, and this is one of the reasons why that action mutation rate is lower than the error rate. Okay? So let's talk about the second one. Okay? So the second big mechanism that contributes to that lower mutation rate is DNA repair. And so, just as a preface to getting into this one, there are actually many different kinds of DNA repair mechanisms in cells, going across all sorts of different types of cells, whether they're bacteria, archaea, eukaryotes, many different mechanisms. It's a very interesting topic. If you ever want to kind of geek out, I highly recommend kind of reading about different DNA repair mechanisms. So we certainly don't have time to cover a lot of them. We're going to focus on one particular form. But again, just remember, there are many DNA repair mechanism cells. Okay? And if you think about that, that makes sense, right? It's really important for all cells to maintain the integrity of their genome, so it would make sense that there are actually multiple mechanisms that are in place to help cells deal with errors that come through and actually fix the DNA and make sure that the right sequence is still encoded in the DNA. So this makes a lot of sense, right? So our focus is going to be on one of these mechanisms. Okay, and we're going to focus on a mechanism called mismatch repair, okay? So this is a mechanism that's actually very highly conserved. Okay? And so this term, I think, carries a lot of weight to it. It's highly conserved. You can think of this as highly conserved evolutionarily. Okay? So this means that there are mismatch repair mechanisms in bacteria that are actually quite similar to the mismatch repair mechanisms in eukaryotic cells and even in human cells. Now, some of the intricate details are going to be a little bit different between that, but this is a fundamental repair pathway that exists almost in all cells on the planet. As we go through our discussion here, I'm going to show you what we know about mismatch repair in bacteria, and that's because it's sort of it's under sort of current investigation in eukaryotes, and there isn't quite as much known about how this functions in eukaryotes as there is in bacteria. So the mechanism that I show you here is going to be in bacteria, so just remember that as we move through this discussion, right? All right, so now the big key with mismatch repair, to contrast it with DNA proofreading here, is that this occurs after DNA replication. Okay? And this will become, I think, even a little bit more important as we go into the intricate details of this mechanism and how it functions, right? All right, so fundamentally, what this mismatch repair can do is it can recognize incorrect base pairs. Okay, so fundamentally not all that different in terms of what DNA proofreading is supposed to do, which is recognize if there are two bases that are in there that aren't supposed to be together, right? And so you can kind of imagine kind of the starting point of how this might occur, because those incorrect base pairs provide some sort of weird shape that can be recognized by other proteins. Okay? So as I mentioned, this mechanism is best understood in bacteria. So let's go ahead and walk through that mechanism in bacteria. All right, so this is mismatch repair. In bacteria. Okay? So this is going to involve a key set of proteins known as mute proteins. Okay, and this mute is actually short for mutant. Okay? So in this case, these proteins were named because of the way their function was identified. Okay, so they were named based off of the consequence when the function of these proteins is eliminated. And that's a very standard way to go about studying the function of proteins in cells. Let's see what happens if we sort of eliminate the function of those proteins. Okay? So in this case, people studying bacteria were able to eliminate the function of these mute proteins, and they found that there was an increase in mutations in these bacteria, which is why they got this name mutant. So this is a name based off of the loss of function phenotype. Okay, so here's a callback to a term we had a couple lectures ago, loss of function. This is a way to describe the consequences of mutations. So in this case, the loss of function phenotype is an increase in mutations, which is why these were then named the mute proteins, right? So let's get into how these mute proteins work. So I'm going to erase this part real here. Okay. All right, so let's say we've got this situation in a bacteria where we have a stretch of DNA, okay? top strand 5' to 3', bottom strand anti-parallel. Okay, and let's say in this case there is an incorrect base pair right here. 5' to 3' there. And we'll put kind of a squiggly line there just to really highlight that this is the wrong base pair. Okay, so this is an incorrect base pair, and this is what the system is trying to detect. And as we've already discussed, these incorrect base pairs, okay, they really kind of serve as a signal because they're the wrong shape. And I've tried to kind of imply that with this sort of kind of bubbling out of the DNA double helix. Now it's not really this. It's much more subtle than this, but this does actually cause a physical change in the shape of the double helix there. Okay. Now this serves as a signal for these mu proteins. So these mu proteins have the ability to detect incorrect base pair shapes, right? So let's put our mu proteins, we'll put them in purple, okay? So these mu proteins have the ability to detect this incorrect base pair, and then they actually bind the DNA right at the site of that incorrect base pair. So it leads to a situation that looks like this. Here's our top strand, 5' to 3', bottom strand, anti-parallel. Okay. Here is our incorrect base pair right there. And what the mu proteins do is they latch on kind of around this whole area. So here's our mu protein. Okay. Now the mu proteins, they actually contain a property, they are endonucleases. Which means they can cut in the middle of a DNA strand. So let's assume in this case that this G right here is the actual wrong base that was added. Okay, so what these mu proteins do is they actually use their endonuclease activity to cleave on either side of the wrong base. Okay, they cut the sugar phosphate backbone right there. Okay, and this allows for this G to be eliminated. But then there's another step kind of after this, and so what I'm going to have to do is I'm going to erase this part here, and we'll continue with this mechanism right over here. Okay. Feel free to pause the video if you want to keep up with the drawings. Okay, so we'll just circle this back to here. Okay. So what this does is it gets rid of the G, and then another set of proteins come in here and actually expand that gap on this top strand. And so this is going to be with the addition of exonucleases. Okay, and we're going to draw those proteins in green. Okay, and what's interesting about this, so we'll go back to our strand of DNA, 5'. There's the G. Oh, the G is no longer there. Okay. Here's our bottom strand. We got the A right there. Okay. Well, these first off, what happens is once this cleave happens here, the mu proteins actually head out of the picture. So mu proteins take off, and then these exonuclease proteins, they latch on here and here. And as their name suggests, they have the ability to degrade DNA from the ends. And so they actually kind of claw back here and here, and they expand that gap, okay, on this top strand. Okay? And that leads to something that looks a little bit more like this. 5', a little bit there, 3', there's our correct base, 5', 3'. So here's this expanded gap created by the exonucleases, okay? Now we have a situation where kind of the protein we all know and love, DNA polymerase, can enter the picture. Okay? It has a free, at the very end of here, remember there's a free 3' OH at the end of that one, so DNA Pol can attach here and simply fill in the gap. Okay? It will likely need eventually the function of a ligase to come in here and kind of really seal that up. Okay? But this is a way that you can eliminate the incorrect base, and as DNA polymerase comes through here, presumably it inserts the right base there. So now we have a correct TA base pair with DNA polymerase. Okay? So this is mismatch repair. Okay? Now, as I go through this, we kind of assumed that G on that top strand was the incorrect base. But you might be asking yourself, how is it possible to know which is the incorrect base? So let's go back to that situation that we described at the very beginning and kind of illustrate this conundrum a little bit. Okay? So this was our starting point. We had our DNA strand, we had a G, let's see, it was a GA base pair, if I remember correctly. 3 prime, 5 prime there. Okay. So remember, this is in the context of a big long stretch of DNA, and this is after replication, right? Right? So if you're presented with this, you know that this is an incorrect base pairing, right? But the question you should be asking yourself is, how does the mute protein know which of these is the wrong base, right? So it could be the G is wrong, it could be that the A is wrong. It really depends on which of these strands is the old strand and which strand is the new strand, right? So this is actually something that the bacteria have a very clever mechanism to help distinguish between the old strand of DNA and the new strand of DNA, and that's what the mismatched proteins can actually recognize, right? So this leads to a really interesting phenomenon called strand discrimination. Okay, and what this means is these mute proteins, they can distinguish between the old and new DNA strands. Okay, the idea here being that the old strand contains the correct base, and the new strand is what contains the incorrect base. Okay? So what is this mechanism? So this is going to get into the weeds a little bit, right? So this is going to get down to the level of chemical modifications of DNA, right? So let's talk about strand discrimination a little bit. Okay, so strand discrimination depends on chemical modification of adenine bases on the old strand. Okay, and the modification that we're actually going to talk about here is what's known as DNA methylation, okay, or the addition of a methyl group. Okay, I want to make sure I'm going to rewrite this one out because it might not be clear. Okay. Methylation, or addition of this methyl group, okay, which in chemical terms is just carbon with 3 hydrogens, or a CH3 group, okay? It's a very simple modification, but what this does is it helps the cells distinguish the old from the new strand, okay? So what might that look like? And again, the key here is that this mechanism, remember, it happens after DNA replication, right? So if we have a stretch of DNA, okay, and this we'll kind of go back to our original stretch of DNA that contains the wrong base pair, but I'm going to put some additional kind of context around it, and that context will include some adenine bases, and that's going to help us kind of illustrate how this process allows these mute proteins to distinguish old from new DNA, right? So here's our 5' strand up here, and let's say, in this case, that there's a bit of DNA, there's an A, and then we get to our kind of G, C, a wrong base pair, okay? And let's say there's another, let's say a T and then an A, 3', and so this bottom strand has this sequence. So again, here is our incorrect base pair, right here. But I've added in some A's and then the actual T base pairs to illustrate that there are some adenines in this process as well. And that's going to be the target of this modification, right? So let's say that this is the DNA, and this is just after actually, wait. No, this is, well, I gotta pause real quick. Yeah, sorry. Yeah. This is not an incorrect base pair. This is a correct base pair. GC is actually correct. Sorry about that. So I gotta take a step back here. So this is correct, okay? And this is just before replication. Okay? So this is before replication, okay? This hasn't been replicated yet. And so this is when this DNA methylation occurs. So just before replication occurs in bacteria, the adenines of the old DNA, so these are both going to be old strands of DNA, are modified with this methyl group. Okay, so I'm just going to put the CH3 there, the CH3 there, and the CH3 right there. Okay? So this, in effect, is the way that the old strand can be distinguished from the new strand after replication, right? So then let's say then DNA replication occurs. Okay, we've got kind of our two strands. We've got kind of strand number 1 here on top and strand number 2 on bottom. And so I'll kind of continue those down here. Okay? And let's say that everything that happens with strand number 1 is just fine, right? So we've got our old strand is on top, 5, A, G, T, A, 3 prime. Remember that these adenines are modified with CH3, okay? And the bottom strand, the new strand that comes in, T, A, C, T, 3 prime. Everything there is good, okay? No mismatch, right? That one's just fine. But let's say that there's an issue with this bottom strand here when it template synthesis, okay? So we'll put this bottom strand down here, okay? T, C, A, T, 5 prime. We'll put this C in orange just again to kind of highlight that this is our base of interest. So there's the bottom strand, and so let's say as the top strand goes through, before we do that, let's make sure we mark that strand has been methylated right there, okay? Distinguishing this one as the old strand in this case. So the top strand, the new strand comes through here, the A goes there, and let's say that this is where we get this mismatch. So let's say in this case that a T is put in place of the normal G, okay? We still get a T there, an A there, 3 prime, and now this is our new strand, okay? And so now we have this mechanism for strand discrimination. The old strand has a methyl group on it, on these A's, and these A's don't, okay? That's what the mu proteins can recognize here. So we have our signal, our kind of incorrect base pair, and now the mu proteins kind of come into the picture, okay? So let's continue on with this one, okay? And so again, DNA replication has occurred, and this mechanism, mismatch repair, occurs after DNA replication, right? So let's continue on right up here. So now we've got 2 things that the mu proteins can recognize. Number 1, the incorrect base pair, and number 2, the methylated adenines. And so this allows the mu protein to assume the right orientation when it actually interacts with the DNA, okay? Because this is the new strand, the mu proteins know that this right here, this is the wrong base. It's the new strand, this is the one that was newly synthesized, this is the one that's got to go, okay? So here's our stretch of DNA, 5 prime, A, T, T, A, 3 prime, bottom strand, T, A, C, T, 3 prime, okay? Because there is a methylation here, the mu proteins bind to this in such a way that they know that this is the wrong base. Okay, so this methylation helps arrange the mu protein so it knows to cleave here. Okay. If this, say, methylation were on this strand, the mu protein would bind in an opposite fashion, in an orientation that would actually cut out this base of the incorrect base pair. Okay, so here's our signal that allows the mu protein to distinguish between the old and the new DNA strand. So then now we go back to the mechanism we just talked about. So now that this has been cleaved, this can engage with the rest of the mismatch repair pathway. Okay, this gets cleaved, mu proteins leave the picture, exonucleases come in, chew back to create a gap, DNA polymerase comes in, fills in the gap, eventually a ligase seals up the sugar phosphate backbone. Okay, so this is the mechanism, this is how it works, and so this is that kind of really detailed look at about how the mismatched proteins can distinguish the old strand from the new strand. Okay, I'll say that this mechanism, this is how it exists in bacteria. In eukaryotes, there's still a mechanism that the mismatched repair proteins use to discriminate old from new DNA. Okay, but in this case, it doesn't have to do with DNA methylation. The current thinking is that it has to do with the leftover kind of gaps that haven't been sealed in the new strand of DNA. But that's kind of what's thought about that, but this is the mechanism of bacteria, right? All right, that wraps up our video for today. Again, please understand that I'm not going to cover this again in a lecture, so this kind of counts as the lecture for today. So please understand this. If you have any questions about this, I'll be happy to talk about it. I'll probably have some review questions in the next lecture or two to kind of make sure that you're getting hold of some of the stuff. So please watch these videos. I will see all of you, some of you in lab on Thursday, and some of you in lecture on Friday. All of you in lecture on Friday. Thanks.